Okay, everybody, welcome back. Going on with Lab 10, more Lophotrochozoans, the mollusks and the annelids. Please bear with us. This is the first time we're doing these recordings online, and hopefully everything works like it's supposed to. If not, send feedback. We need to make sure this works as well as possible for you guys. So, mollusks. Epimorphs for the mollusks are going to include a radula, calcareous spicules forming a shell, a muscular foot, and a mantle. This is a generalized diagram, and so this is not any specific mollusk, but these are all the kind of the general things that a mollusk is going to have. You'll see the mantle. Radula is this rasping tongue-like organ, scraping things into the mouth. We'll get to more in a second. Moving on from the mollusks. Anyway, apomorphes, mollusks, they're all going to have this to some degree. Basal lineage of the mollusks, we got the polyplacophoras. We've got local ones here. We've got chitons. If you've ever been to the tide pools, you'll see them stuck onto the rocks. Identifying features for them are going to be 7 to 8 separate articulating shell plates, which you can see here. Right? These are individual shells, but they can move. There's kind of like a little bit of a fleshy joint in the middle. And the mantle is fused to the shell plates. Moving on from them, we've got the cephalopods and the gastropods. Both develop a well-developed head with sensory structures and are united by the evolution of a coiled shell. However, but some of them have lost or reduced in some species. Right? This is going to be things like your squids and your octopus and your snails and your slugs and nudibranchs. So the gastropods, one of their big things, especially with reference to the snails, they undergo a process called torsion, which is the rotation of the visceral mass. So their gut and nerve cords are twisted. So you want to note the position of the mantle cavity and the anus. Basically, if you've evolved a shell, right, and your back end is stuck into a shell, you can't poop into your shell, right? So your visceral mass has been twisted around, and now your butt is basically, you're pooping on top of your head. Not the most pleasant experience, but, you know, they're a snail. They don't care. The nudibranchs, right? These things are super cool. Right? Nudibranchs, we've got some for you to look at in the lab. Sorry, pictures for you to look at in the lab. Right? They have lost their shell and undergone secondary detorsion. So in their evolutionary history, they had a shell and they had that torsion where everything was twisted around. Once they lost that shell, they're able to detwist themselves, right? So the elids, right, they eat cnidarians and pass their stinging cells from the digestive system to the tips of the feathery serrata. These are the ones I was talking about where we looked at last week, all the cnidarians, things with those stinging hydras and the corals and the sea anemones. Well, they eat these stinging cells, and they pass them up into their skin, and basically use them as their own form of self-defense. The dorids, they prey upon the noxious sponges and incorporate the noxious compounds into their epidermis. So you have the elids, which they basically eat bullets and put them into those feathery serrata, these things flaking off their back, and use them as defense. And the other ones incorporate all the toxic compounds that they can't make themselves, but they put it into their skin, basically saying, "Don't touch me." And how do they advertise that? They have these aposematic coloration. This is bright colors as a warning to predators. So if you're trying to blend in, you don't have a lot of defenses, and that's your main defense. But if you're advertising your presence, you are fluorescently colored, screaming, "Look at me." It's a warning saying, "Don't touch me. If you eat me, it's going to be a bad day for you." So aposematic coloration, also known as warning coloration, is a pattern you can see throughout animals all over the world. Think poison arrow frogs, fluorescently colored. The cephalopods, right? Their name literally means head and feet, right? Cephala, head, poda, foot. They are head and feet, right? Only the nautilus has an external shell, right? This is a really cool little dude here, right? It's a septate shell. Remember the word septate? So it's got broken into chambers with divisions, and a siphuncle to control buoyancy. Right? Unfortunately, we don't have one of those to look at, but they're super cool. The more common ones that you're more familiar with are going to be the octopus and the squids. For them, their foot has been modified into tentacles and arms. They have a beak-like jaw, which it's called the beak-like because it looks like the beak of a raptor. They have a siphon that is used for propulsion, reproduction, and ink release. The ink sac confuses the predators. So whenever they shove off, you'll see they can releases bursts of, basically have a smoke screen. And they have a fusion of ganglia, right? So they don't have a brain proper, but octopus can be crazy smart, and I can tell you about some other things about how cool and smart they are. But moving on from that, so hopefully this video plays in real time. All right, so what we have here, right, this is just to highlight the ability of the octopus to mimic its environment and camouflage itself. This is real time. This has not been delayed or modified in any way. And that is an inkjet you just saw. All right, they want to get out of there, so they go hit that jet propulsion. So now we're going to slow motion it and put it into reverse, and you can see just how rapidly they can change their skin color and texture to match the surrounding environments. All right, so we have to slow it down to see just how fast they're latching on to a rock that's got some algae and some other things on there, and they go, I don't want to be seen. Boom. You can actually see some of the, if you look at the skin, it changes texture, which is super cool. So moving on, if you can see in the upper left-hand corner here, so these are just different examples of some of the cephalopods, primarily the octopus, and how they can change and adapt to their surroundings to blend in. So this is the video we just saw, kind of broken down. Some of them have bioluminescence when they live really deep. The one on the left in the middle, right, this is one species of octopus, right? He's blending in to look like a flounder. This one's trying to make itself look like the thing on the right there, lionfish, venomous lionfish. And this one is moving so it looks like a sea snake. Right? They're really adept and very intelligent, like crazy intelligent. For the squid dissection, which we'll have a video for you guys to look at online, what you want to be paying attention to are the ones that I've highlighted here in red. Right? You look at the funnel, and they got the arms and the tentacles. So the arms are the short ones. They got lots of those. And they got suckers all the way up and down the arms. The tentacles only have the suckers at the very end. Right? They've got an ink sac in there. And if it's a male, it's going to have the testes up here. And if it's female, it'll have ovaries up there. And the ctenidium, right, also called gills. I'd accept either answer because that's exactly what they are. Moving on from there, we've got the bivalves, all right, bi-two valva meaning plates, so it's two plates, right? So think of mussels and clams and oysters, things that have two plates. They've got a reduction of their head. Their shell is composed of two plates, like I just said, and they are filter feeders. Their gills have been modified for filter feeding. They still use their gills for breathing. Their gills, that's what gills do. However, they've also got really good examples of them, we'll show later on, that they can use them to filter feed, and you'll see these little ciliated membranes that can just move the particles a lot. And they have lost the radula, though. Right? They have, along their evolutionary track, their radula is gone. For the clam dissection, what you're going to be looking at here is we've got the gills, are these, can't see that? So anyway, the gills line the shell. So the big fleshy bit, for like most mussels, what you'd be eating is the mantle. The mantle is what excretes the shell, right? And it just keeps building on itself, which is why they just keep getting bigger and bigger. And the gills are going to be these very light, feathery structures on the inside. And when we do when you watch the video of the mussel dissection, you'll be able to see, zoomed in, how they can use those gills to move particles up and then use that as for filter feeding. The gills, for the filter feeding, they push everything up along to the very top edge of the gill, and then it gets pushed down here into these palps and into the mouth. The palps basically just ball up that mucus ball full of all the food particles and shove it into the mouth. And the abductor muscle is here up on the top, and that's basically what just keeps the shell closed very, very tightly for the mussels or clams. Moving on from there, we go into the annelidas. Right? These are not mollusks, these are different phyla. So we got the annelids. Apomorphs for the annelids, they have a segmented body plan. All right, so they're coelom, partitioned by septa. Right? They got prostotum and peristome with a mouth. That's going to be this section up here. Right? So remember the stoma. Right? So they got before the stoma and after the stoma and around the stoma. So peristome with the stoma in the mouth. They have epidermal setae. So these are bundled pairs, segmentally ranged, bristle-like structures with aid locomotion. It's a really long handful, but if you've ever held an earthworm in your hands, you can slide from the head to the tail, right? Tip of the tail in one direction. It's very smooth. And if you go reverse, it's a little rough, right? Those are the SETI that are just, it's pushing its way through tunnels, and it's got one directional barbs that help it push. The polychaetes, right? Polychaeta, they are come in two forms. You have free living and two dwelling forms. Epimorphies for them are going to be the parapoda, right? So these are unjointed appendages that aid in locomotion and respiration. So all the little things sticking off the side are going to be the parapoda. And then the head is elaborate with sensory appendages. That can mean either little, tentacles that feel stuff, or antennae. Not necessarily going to have eyes. I don't think any of them actually have eyes. But sensory appendages, but not always eyes or nose or anything, but just help them feel the world. So we've got free living forms that, they're free living, they do what they want, versus tube dwelling forms. So they're tube worms. They come in everything from millimeters to meters long. Those are the very deep oceans. And so, yeah, those are your polychaetes. All right. I've posted a couple of videos. This is probably one of my favorite polychaetes. This is collectively commonly called the bobbit worm. So the Unis aphratoides, right? And injects toxin into their prey, stuns or kills. Just watch the videos. It's super cool. Also called a sand striker, but they're very, very lovely and terrifying. Moving on from the polychaetes, we have the oligochaetes and the hirudinias, right? They are united by the fact that they are hermaphroditic. So individuals can be both male and female. They have a clitillum, right? So if you've ever, it's a swollen glandular region, functions in reproduction. So if you're ever looking at an earthworm, just a standard old earthworm, and you see a thick band, let's see if I can highlight that. Okay, so I've tried to circle that. Hopefully you can see that. That's the clitillum. That's just one of the epimorphies used in reproduction. They have lost the trochophore larval stage in adaptation to terrestrial environment. Remember, the trochophore larval stage was these little free swimming, swimming around the marine environment. But if you live on land, in the dirt, not very useful to have a free swimming larval stage if you live in dirt. The hirudinias, epimorphs for them are going to be the posterior body sucker, right? And then a subdivision of body segments by superficial annulae. This is a leech, right? Yeah, they're leeches. So they got suckers at both ends. So posterior allows it to kind of inchworm along if it's trying to move up an object, and you guys how it latches on. But its main sucker mouth is going to be where the mouth is, and it's a leech, drinks blood. The annelid muscle movement, right? This is peristaltic movement. It's got circular muscles and longitudinal muscles. The diagram it's much easier to see, so I'll post a video and you can see it moving, and it moves in these accordion-like sections. And as the longitudinal muscles contract, it brings it up, and the circular muscles relax, so it gets shorter and fatter. And then as the circular muscles contract, the longitudinal muscles will relax, and it helps it squeeze itself forward, right? And it pushes it forward. Think of squeezing like a water balloon, or if you've ever milked a cow, it's that peristaltic movement. It's also the same type of movement that pushes food through your digestive tract, right? And that's how they do horizontal movement. And that's it. Okay, so you guys, make sure you look at the videos and some of the pictures that are going to be posted online. Hopefully, you'll be able to hear me very well. Please give me feedback in email if you can't hear something, or if I need to slow down or what have you. But like I said, this is a work in progress, so let us know how we're doing, and we'll get through the rest of the semester, and we'll do our best to make sure this doesn't affect you guys any more than it has to. Stay safe and wash your hands. Take care. Bye.